Hi, everyone. Welcome to episode 121 of Cell and Gene, the podcast. I'm your host, Erin Harris, and this episode is a big one. I'm joined by Benjamin Oakes, co-founder and CEO of Scribe Therapeutics, for a conversation that challenges how we think about genetic medicine, moving it beyond rare diseases and into the realm of common chronic diseases such as cardiovascular disease. Ben and I talk about building safer, more precise CRISPR tools, what it really means to design genome editing for millions of patients, and why Scribe is betting big on durability, prevention, and long-term impact. We get into the science and the strategy and the big picture vision behind Scribe's push into cardiometabolic disease, and why Ben believes genetic medicine should be built for millions, not thousands. And so here's my conversation with Scribe Therapeutics' Benjamin Oakes. Enjoy. Ben, hey, welcome back to Cell and Gene, the podcast. I'm happy that you are here to talk to us about what's been going on at Scribe Therapeutics.
Yeah, Erin, thank you so much for the invite. We are really pleased to share a bunch of updates this year, looking into, I think, a very exciting next year.
Good. Well, we're going to get into it. So talk to us a little bit about Scribe Therapeutics.
Yeah, so I'd say first kind of level set everyone on Scribe. If you're unfamiliar with us, we were founded and built as an organization that has really been focused on using high throughput and holistic molecular engineering, which basically just means really deep, thoughtful, and intentional tool building around genetic medicines as a whole, with the explicit goal of trying to figure out and then build the technologies that allow us to take genetic medicines from the 1 in 100,000 or the 1,000 rare disease patients, if you will, to 100,000, a million, 10 million common disease patients. So actually, to be even more explicit, when I sat down and co-founded Scribe Therapeutics with Dave Savage and Jennifer Doudna, and we really asked, where should genome editing, genetic medicine as a whole go in the future? And what we realized is that it shouldn't be relegated just to rare disease and ultra rare disease and just to areas where you may have a monogenic disease. In fact, we wanted to try to figure out how we can bring genetic medicine to everyone, right?
Mm-hmm.
How can we, and if we do so, can we essentially kind of transform how we think about medicine, and potentially in doing so, even transform how we think about things like healthspan and even longevity. That is the promise of being able to modify or change or kind of tune your genome. But the tools and technologies that were first developed and are actually in widespread use today, we didn't believe were kind of up for the task. So we've focused in on that, really tried to develop technologies that are both dramatically safer and more effective to enable essentially this application in everybody.
Good. Thank you. I'm glad you brought up your partnership with Jennifer Doudna. We're definitely going to get into that later on in this conversation. Before we do, I want to talk a little bit about your work with cardiovascular disease, the number one, disease worldwide, the number one disease.
Yeah.
Talk to us a little bit about how cardiovascular disease in general has been an opportunity for you and your team to make the biggest difference. And what are you and your team doing to address the limitations that cardiovascular disease, the current treatments, provide the patient?
Yeah, no. So this is a really, I think, important point. You kind of touched on it already. I'm gonna hammer on it some more, which is that cardiovascular disease is the leading cause of death globally. It outstrips cancer almost 2-to-1. Yet you hear about things like cancer, Alzheimer's disease and dementia, a lot more. And that is unfortunately, I think, because of a number of reasons, but cardiovascular disease is a silent killer for the most part until you have, a pretty catastrophic event. We chose specifically to work on cardiovascular disease from the very early days of Scribe, in large part because it is this area of kind of almost deceptively the greatest unmet need, right? It flies under the radar despite having the greatest unmet need. And then also, I think it's near and dear to many of our own hearts. And I mean that like literally. Uh, I talk very often about how my family has been affected by cardiovascular disease and how
Mhm.
the joke at Scribe in many ways is we're building a lot of our drugs for Ben. Um, so
I
it's
don't mean to laugh. It's not.
no, it's
Yeah,
not, but it's also, I think it really underscores what we're trying to do and how it's dramatically different than
sure.
what almost every other company in the genetic medicine kind of CRISPR genome editing space has done, which has been kind of this pick up a technology from academia and apply it as rapidly as possible. We don't have that viewpoint on things. Our goal has been, okay, what are the diseases where we think genetic medicine can have the greatest impact for the most people? ASCVD, cardiovascular disease, is really number one up there.
Mhm.
And how do we build the tools and technologies that are best suited for that application was the question at Scribe since the early days. And now it is really how do we actually expand from those initial applications even into a number of other applications in cardiovascular disease. So kind of unpacking that one layer down, the next question is, well, like cardiovascular disease, if it's such a large disease, if we knew how to solve it, wouldn't we do it already? Like why have we not solved it already, just despite its importance? And I think the interesting thing about this, and one of the most important reasons why I believe very highly durable genetic medicines are going to play a role, is that cardiovascular disease and ASCVD in particular is a lipid accumulation disorder, right? It is an accumulation of lipids in your arteries, and we actually fully understand what those lipids are, right? You can kind of think about them. I think about them in three simplified buckets, of course, but you have LDL-C, or what's, colloquially known as bad cholesterol.
Mhm.
You have this other particle that's becoming a lot more interesting to many people, known as LP little a, or lipoprotein A. And then you have remnant cholesterol and triglycerides. And those three buckets make up the majority of the lipids or the cholesterol that most bodies produce. And everyone actually has a different level. Those three things together are, as I think about it, the majority of the gas in the tank for something like ASCVD, which is really interesting because we now have three very core targets that we can go after. And the genetic data that exists shows that if you can modify one, even just one of them, or potentially multiple of them together, you could reduce incidence rates of things like heart attacks by greater than 50%, maybe even as high as 90%. The problem, Erin, is that despite having drugs for these lipids, things like statins, things like bempedoic acid and ezetimibe, things like even PCSK9 inhibitors, most patients can't stay on those drugs for longer than a year or 2 years. I think the
Mhm.
average the average patient stays on statins for no longer than 12 months. And lipid accumulation is a lifelong accumulation. You need to modify lipids for decades to really have the effects that, of course, those genetic studies that I said could reduce by up to 90%, that's someone who has lifelong lower lipids. So to achieve that essentially near curative state for ASCVD, you need to be able to recapitulate that lifelong state. The only way, in my mind, I think, to do this is to provide genetic medicines. And I think that is one of the real opportunities we have to really dramatically shift the curve on cardiovascular disease, which would have a huge impact on human health overall.
Yeah, absolutely. That's really fascinating information that I don't really, at least me personally, didn't quite appreciate or understand those numbers in the way that you're describing them. So that's fascinating. And I think, and I suspect we'll get into this next one because I want to talk a little bit about the trials that Scribe is in the midst of. And when they came across my desk, so we're talking about STX 1150, 1200, and 1400. We'll break
Mhm.
those up. So I guess let's start with STX 1150. And that showed more than 16 months of LDL lowering from a single dose in primates.
Yeah, that's
Talk
right.
to us a little bit about that. What did you learn in terms of durability, dosing? You mentioned, excuse me, PCSK9 therapies. Talk us through that trial a little bit.
Yeah. Yeah. And so I think the easiest way to kind of understand what Scribe is trying to accomplish right now in terms of our lead assets is to build assets that can modify each one of those major lipid risk factors. And like I said, many patients have an elevated level of just one. Some patients have elevated levels of two of them or three of them. So those lipid risk factors, again, are LDL-C, LP little a, and triglycerides and remnant cholesterol. Our lead product is, as you mentioned, is STX 1150. And that is a product we're very excited about bringing forward for LDL-C lowering. What's really important to kind of understand about this product is that it is an epigenetic silencer that does not cut the genome at all. So we build CRISPR genome editing therapies in one instance, and another set of technologies we've built is epigenetic silencing therapies that do not permanently modify the genome or the genetic sequence in any way. And we think that the epigenetic silencers are actually really fantastic tools to be utilized on risk factors like LDL-C, which are in many instances strongly genetic. I can say, like, I have done as much as I possibly can from a lifestyle perspective, and my LDL-C, for example, doesn't get low. So there's a large genetic component, but it's not a monogenic component, right? I don't know what genes are broken inside me to fix. And there are lifestyle and other types of interventions. And for that sort of really multifaceted, complex risk factor modification, we really like the epigenetic silencing approaches because we do feel like they are going to be much more attractive to both patients and doctors. So while we had built genome editing tools to lower LDL-C, we actually chose to advance, and we'll be advancing next year into the clinic, our lead program for LDL-C lowering via epigenetic silencing. Now, as you referred to, we just released some updated data at the American Heart Association conference that is very exciting, which I believe is the first demonstration of epigenetic silencing of PCSK9 to lower LDL-C, achieving greater than 50% LDL-C lowering at, very critically and importantly, a therapeutically relevant dose of less than 1 mg per kg of LNP, right? In the field, there's been some challenges with LNP delivery, and therefore we really want to keep those doses low. The thing that is even more exciting is we are now, with a non-permanent genome modification therapy, at over 515 days in these non-human primates with no sign of losing LDL-C reduction. And this kind of gets back to the conversation we just had, which is how do you build a therapy that can last perhaps years or decades, that doesn't increase patient burden, that someone could essentially get on and never have to worry about their LDL-C again? We think that those are the types of therapies that will be necessary to change the outcomes in ASCVD even more dramatically than some of the more recent studies, for example, with PCSK9 inhibitors. But the field itself, I think, is also moving forward quite rapidly towards these more effective PCSK9 inhibitor style drugs.
Excellent. Okay. Similarly, but moving on to STX 1200 and 1400.
Yeah.
Again, came across my desk, both delivered very deep target reduction with no detectable off-target editing. So talk us through how Scribe's engineered CasX platform achieves that level of precision and the why behind it. Just kind of bring us up to speed on what's going on there.
Yeah, so that's a great question. So again, to kind of frame for the audience here, the 1150 program is an epigenetic silencer for LDL-C lowering. 1200 and 1400 are genome editor therapies. And we really think about using a genome editor when there is a very well understood genetic cause for elevated lipid. And that cause is either monogenic or at least well understood to be able to be modified by a permanent genetic cure, right? So, and this is actually very interesting. This is the case with LP little a, where elevated levels of LP little a are almost entirely determined by your genetics, greater than 80%, in some studies, actually greater than 90%. And therefore, it really is a genetic disease. And actually, elevated LP little a likely should be considered the largest genetic disease that currently exists. So we have built a genome editor for elevated LP little a. We have demonstrated in transgenic mouse models greater than 90% reduction, actually greater than 95% reduction of this protein after genome editing of the liver. And we're very excited about that because that's on par with, if not actually exceeding, many of the RNAi and ASO style therapies that are currently in development for that target. Of course, we're able to provide that one and done sort of durable outcome for patients that we think is going to be really transformative for the field of ASCVD. Likewise, just skipping over to 1400 before I get back to how we've engineered these things to be as safe as they are.
Sure.
1400 is a therapy actually for elevated triglycerides. And the development path for that is a little bit different. It is not directly focused on ASCVD initially. It actually starts out in a rare disease known as FCS, or familial chylomicronemia syndrome, that is, I think, a really important target for elevated triglycerides because these folks have essentially no other way to modify their triglyceride levels except for now these ApoC3 targeting therapies that others, there's actually been 2 approved now in the past year that are very exciting. Still, those are RNAi style therapies that need to be delivered, monthly or quarterly. And we want to provide those rare disease patients with a really highly durable, highly safe cure or near curative therapy, if you will, for that disease. I think from there, it becomes a very interesting question if triglyceride lowering can be used in patients who have elevated risk of ASCVD and elevated triglycerides to help lower basically your cardiovascular event rates. And we'll, of course, have to study that when the time comes. But both of these therapies have shown, again, greater than 95% reduction of their target proteins. In animal models of disease, we actually released some non-human primate data in the ApoC3 program for triglycerides demonstrating that we get, again, complete genome editing below 1 mg per kg. And we're really excited about that level of potency coupled with the really important point, Erin, you mentioned, which is, again, something that we have not really seen in the field from others, which is a level of specificity where we are unable to identify off targets even at like really maximum supersaturating doses. And so this is a bit complicated, but the primary concern, I think, of genome editing tools in the past has been these off-target events. And this is really interesting and makes sense because, quite frankly, genome editing tools, quote unquote, are not really genome editing tools. They're bacterial immune systems that were co-opted from nature and then applied fairly rapidly. As I mentioned earlier in this episode, Scribe has never been focused on kind of taking that bacterial immune system and just using it. We have spent, now years, if you will, kind of melting down and reforging these bacterial immune systems into what we want, right? And so the key thing to remember is that when a bacteria or a microbe kind of evolves these CRISPR systems, they're using it as an immune system that has certain characteristics. Those characteristics are by no means the same that we want when we use them in a human to edit only a single location. And I think the most interesting point here is that, as I think most people can understand, if an immune system is perfectly specific, if it only can target one virus that has the perfect match to its genome, that immune system is going to get evolved around very quickly. Right? And this is why our immune systems are so adaptive and why actually a lack of specificity is built into most immune systems. Well, it turns out that's actually also the case for CRISPR. It's not good to be perfectly specific if you're a CRISPR bacterial immune system. Contrary to that, we need to be. So we've essentially put our technologies through these iterative engineering cycles where you're finding essentially characteristics that don't exist in nature. They should not exist in nature, of really exquisite specificity. And by doing that iteratively time over time, while also at the same time selecting for potency, we've been able to generate technologies that are, improved orders of magnitude in terms of potency over where they started out in nature. And at the same time, do not edit at off targets, even at supersaturating doses of 10x. And this is what we've now shown. We've shown this for the 1200 program prototypes. We've shown this for the 1400 program prototypes. And they, again, coupled with the potency we're seeing in animal models, I think is the first example of having both really extreme and extremely exquisite on-target potency without having that off-target genome editing risk. And the question becomes, since we continue to show this, at some point, should the off-target risk for at least platforms like ours at Scribe really not be as concerning, right? If it does not occur, we think that's a much better profile for treating, again, thinking about treating, hundreds of thousands, millions, potentially tens of millions of patients.
Gotcha. I want to talk a little bit about partnerships, and rightfully so, Scribe has already attracted major partnerships around your platform. And so if you're able to talk to us about who those partners are and why you're collaborating with them, and what you've learned from these collaborations that you're able to apply to your own internal programs, or perhaps even just best practices for the listener when choosing a strategic partner.
Yeah, so that's an interesting it's an interesting question. I think there's a lot of parts of it maybe I'll try to break up. The first is, so Scribe is very fortunate right now to be have some very productive and active collaborations with our friends at Sanofi. That started out actually as a ex vivo collaboration and then expanded into an in vivo collaboration where we're going after the underlying cause of sickle cell. And then we also have expanded to another genetic disease as well. Our second partnership is with our friends at Lilly, where we're really focused on neurological and neuromuscular diseases. And in both of those instances, what we have found, or really what we sought out, was to work with partners who had technologies that were real complements to what we were building at Scribe, which is, these really exquisitely potent and specific genome editing tools and epigenetic silencing technologies. Both Lilly and Sanofi were bringing their own really unique takes on delivery to those partnerships, in what allowed us to kind of come together and accomplish drug creation in a way that neither of us could have done alone. I truly believe. So we're really fortunate also at Scribe in being very focused on our cardiometabolic pipeline. I think we've seen the error of trying to do everything in the CRISPR space. There's been many organizations, really in the genetic medicine space, where they just get over their skis in terms of, dozens of pipeline programs across dozens of different diseases. And that requires dozens of expertise levels that are just very difficult to build out. We, from day one, have been really, again, very intentional about having a more disciplined and focused approach, first on engineering and then on application of our technologies in an area where we can leverage an expertise we're building, which is, cardiometabolic and really cardiometabolic disease. Outside of that allows us to find the right partners who can bring their expertise, both disease as well as potentially delivery, to the fore and help us advance things. So earlier this year, we did announce a milestone with Lilly in one of those programs. I believe just before that, we announced a milestone with Sanofi and kind of advancing some of those programs. So I'm very excited about where that work is and where it will continue to go. And at the same time, we're always looking for more ways to apply our technologies that we won't actively or can't actively pursue today, right? I truly, I think this probably comes across, but we truly believe at Scribe, everyone should get access to the very best of genetic medicine. It should be available for every disease that it makes sense, which quite frankly is most. I do think that in the future, the majority of medicine will look like genetic medicine. And we can't do that alone. So we're always actively looking for folks who can bring their own insights, their own delivery, or their own kind of unique take on things to the table, and we can enable with technologies that fundamentally appear to be dramatically safer, dramatically more potent, and therefore can kind of unlock that next level.
Sure, better together makes perfect sense. I want to round out our discussion here with, so at the top of our call, you mentioned Jennifer Doudna, who of course you co-founded Scribe with, and you co-founded Scribe at a time when CRISPR was really like we say here, a rare disease story. And so what convinced you and Jennifer and your team that cardiometabolic disease should be the focus? And I guess on top of that, how do you see your CRISPR by design approach expanding into other common diseases over time? As you said, genetic medicine should be, and hopefully will be available across the board in the future. So, what are your thoughts?
Yeah, no, this it goes. Yeah, this is a it's an interesting question because it goes back to my like fundamentally what I think I thought I wanted to do early on, right? Um, should probably be as no surprise. I took the MCATs. I thought I was going to go become a doctor, and at the same time, when I started working at hospitals, I realized that a lot of medicine was more focused on stabilization, on treatment of symptoms, and treatment very late. And that sent me down this path, close to 15 years ago of looking for what the future of medicine should look like. And at that point in time, there was no really CRISPR genome editing technology. So I started working on zinc finger nuclease technology. That was the first. It was the first thing has actually been around, since the mid 90s for doing genome editing. And when Jennifer so like that's where I come from is this deep desire to get to building a medical system that treats underlying causes, right? And I think obviously when Jennifer showed the world how to use Cas9 in 2012, I immediately switched from zinc fingers and I came out to UC Berkeley to work with Jennifer, as well as Dave Savage, another professor who's more focused on protein engineering at Berkeley. So when I graduated and I was given this really fantastic opportunity through the Innovative Genomics Institute Entrepreneurial Fellowship to start my own lab at Berkeley, I sat down with Dave and Jennifer and we really had this discussion about where should the future of genetic medicine go. And what we realized is that, look at, yes, Cas9-based therapies, base editing, that sort of stuff was probably sufficient to treat rare disease where risk benefit was maybe more acceptable, where you could deal with some off targets because the risk of the disease is so huge. Mm-hmm. But what we also realize is that in order to unlock the future of medicine as we see it, which is we should all be treating the underlying causes of our disease before we get them, so we don't get them in the first place. Not this like, okay, I'm going to wait till I get disease. I'm going to show up at the hospital. They're going to cut me open. They're going to crack my chest. They're going to move a bunch of arteries around, and then they're going to put me on statins, which is like standard of care today. In order to transition to a different model of healthcare, we would have to transition the technologies from these, maybe riskier technologies to ones that have much lower risk levels. When you think about, again, where that application makes the most sense, because of our understanding of lipids and because of a lot of the genetics that have been done around cardiovascular risk, actually, cardiovascular disease is not only the largest, as we talked about at the beginning, but it's also a disease where modifying only a small number of pathways, right? We've talked about lipids. You could talk about blood pressure. You could talk about things like obesity and insulin resistance as well. Mm-hmm. And then finally inflammation. But a very small number of pathways, if you modify them effectively, you can treat this disease, which looks very different than other rare disease, looks very different than cancer, looks very different than a number of other pathologies that are yet to be figured out in a simple way. So coupling genetic medicine to cardiovascular disease made a lot of sense to us if we could use engineering to kind of bridge that gap from the safety necessary to treat 1,000 patients to the safety necessary to treat 1 million patients. And that is where we ended up. That is, I think, what makes the most sense. And then, as I also already mentioned, I think it's quite clarifying to think about what drugs you would want to take. And I think that's also something that's unique to Scribe, is that the drugs we are making very often for ourselves or our family members.
Sure.
And that is a different, I think it's a different level of motivation.
Well, that wraps up this podcast, this episode of Cell and Gene, the podcast featuring Scribe Therapeutics, Benjamin Oaks. Ben, thank you again for your time. This has been a real delight.
Thanks, Erin.
Thank you, listeners, for tuning in, and be sure to subscribe wherever you podcast so you never miss an important episode just like this one. And we will talk to you soon.